The overall goal of this procedure is to demonstrate the dual labeling of Kaposi's sarcoma tumor tissue by immunohistochemistry for viral and host cell proteins. This is accomplished by first exposing antigens using heat. The second step of the procedure is application of primary antibody conjugated to biotin. The third step of the procedure is the addition of the strepavidin-enzyme conjugate. The final step of the procedure is the addition of the enzyme substrate to the tissue for color development. Ultimately, results can be obtained that show the localization of both viral and host cell proteins in the tumor tissue through immunohistochemical staining.
This method can answer key questions in the field of pathology, such as identifying infectious agents as well as dysregulated cellular genes in the same specimen.
Under a fume hood, place up to 8 chemate slides with 5 micrometer sections of fixed paraffin-embedded tissue in a Kaplan jar of xylene. Incubate for 10 minutes to deparaffinize and prepare the slides for dual-labeled immunohistochemistry. Rehydrate the tissue in a graded series of 100%, 95%, and 70% ethanol solutions for 5 minutes each. Transfer the slides to water for at least 10 minutes before antigen unmasking, also known as antigen retrieval. For antigen unmasking, submerge the slides in a plastic container containing freshly prepared 95 degrees Celsius 50 millimolar citrate buffer. Heat in a microwave oven to maintain the boiling at 95 degrees Celsius for 15 minutes. Immediately transfer slides to a Kaplan jar full of distilled water at room temperature. Transfer slides to a clean Kaplan jar filled with freshly prepared quenching solution. Incubate at room temperature.